So, first of all, I want to thank Austin. Austin and I have bumped into each other over the years a lot, and he kind of stole some of my thunder because he raised a number of the issues I was hoping to bring up that I thought hadn't been raised here yet. So you're going to see some overlap. In general, okay, first of all, I'm from Hawaii. Hawaii is one of the most expensive places to work in the world. It's one of the most isolated. Our reefs are, I mean, we're an outlier. They're weird compared to everywhere else. We don't have a crop rate in any sense of the word. Our corals are extremely slow growing, 1 to 2 centimeters a year on average. So it forces us to move in very different directions than most other restoration efforts have done. And that's caused us to develop things slightly differently than others. But in general, we can look at two different ends of a spectrum in terms of reefs, and corals in general. You have the extremely fast-growing crop-rid reefs where you get lots of branching species, and a lot of the images you've been seeing over the last couple of days have been doing this kind of thing. And that's great. There's nothing wrong with that. These are imperforate corals, meaning that the tissue exists on the outside of the skeleton, and that type of coral is very different than massive corals and some of the other forms of corals, which are imperforate corals, where the tissues extend way down into the skeleton. Ecologically, they're like apples and oranges. And we haven't been really treating them that way. These imperforate corals trend very well towards in-water nurseries, these fast growth techniques you've been hearing about. Slow-growing corals, though, don't. And when you get to a place that's in very cold water like Hawaii, with extremely slow-growing corals, 1 to 2 centimeters a year, your techniques are very different. That said, those aren't the kind of reefs we want in most places in the world. We want reefs that are made up of a blend of corals. We want the digiforms, the tables, the columnar, the plate, the branching, and the massive corals. We want those assemblages, and we want the species diversities that are associated with them. And what we need to be thinking, I mean, it was great that most of the efforts have focused on fast growing. Now we're going to start thinking about how we apply things to grow all the other types of corals, and not just hard corals. I mean, there's been very little discussion here about soft corals, sea fans, corallomorphs, zoanthids, large sponges, all these, and crustose coral and algae there's been a little bit of, but these are all the components of a reef, and we need to be considering that if we're going to be talking at the scale of ecological restoration of reefs. So from our perspective, we're at extreme, we do things a little differently, and just to kind of move you in a different direction, you know, if I were to ask you really quickly, what would you prefer, 90% coral cover on your reef or 15% coral cover? Most of you are going to shift and instantly say 90% cover. But if I were to say it in another way, should I be producing thousands of coral frags, small coral frags, or just a couple really big colonies? Most of you would still say produce lots of the small coral frags. All right, so coral cover is like plant cover, okay? That's 90% plant cover. That means everything you wanted. I've got, as far as you can see, there's plants. All right? That's 15% cover. Very, very different. They're both plants. And actually, to be fair, I mean, if it wasn't grasses, we could say small plants will eventually grow into big plants. So I'll gain those services and functions that that big tree has, but the difference is what your time frame is. But so far, we've been mostly focusing on putting out lots and lots and lots of small things. And we got to keep in mind that these big things provide very dramatic services and functions. And how do we get there? So size matters. Most animals have, actually animals and plants have something called a size refuge. It's a very important ecological concept. A number of practitioners here, I can see 3 of you instantly that I know are using this because you're targeting putting out corals at a size of reproduction in order to take advantage of some of this. These types of nurseries are in-water nurseries. People have been talking about a lot at this meeting, and they're great. They work extremely well with the types of corals we've been talking about. But you're going to be hearing from Vaughn and myself and maybe 1 or 2 others about different types of nurseries, focusing more on the slower growing corals, the perforate corals. And you're going to be hearing about this term called microfragmentation, where we kind of trick the coral into growing fast by doing a number of things, one of which is by making it really small You increase that surface area to volume ratio, which allows it to grow faster in all directions. There's also some thought that you're basically turning on a stress response, so you're going to get them growing faster that way. But at these extremely small sizes, they are prone to stress from other variables, so they require some care. And here's what we're doing in Hawaii. We're doing the microfragmentation. We are putting out modules that are fully covered. And actually, it's one of our tenets. We don't put out things that aren't less than 100% covered when we put them out in the field. And we have to do this, as I said, because of these conditions that I talked about earlier that are affecting, make Hawaii an outlier. So techniques like this really don't work well in Hawaii. The in-water nurseries haven't worked, and it primarily is due to that cooler water, slow growth rate. So when we went to learn to think about how we were going to produce a nursery, I'm a regulator. I manage reefs for the state. I deal with a lot of researchers, but I also deal with aquariums. And I was dealing with some aquariums and talking with some people on the U.S. mainland who were dealing with aquariums, and found out that for decades there have been guys in garages in the middle of the Midwest growing corals, fragmenting corals, and because it was difficult to move material around, had been sharing it in small networks, and they kind of stumbled upon the same thing that years later we did. In addition, there were very, very large aquarium trade companies that had developed these huge warehouses, many of them in places like California, where they were fragmenting, developing raceways, specialized light systems, specialized water systems, and talking with them, we were able to modify these techniques to work for resource management. But we have a problem because we're dealing with a slow-growing coral, and with a fast-growing coral, I can go break off a piece, and the idea is it'll recover fast enough so there's very little, if any, impact that you can measure in the natural environment. With a slow-growing coral, if I take a coral from here to bring it into my nursery to go then restore an impacted reef, I have that impact on the area where I collected it from. So we needed sources of corals that wouldn't cause impacts. And luckily in Hawaii, we actually have a lot of coral growing in our commercial harbors and our recreational harbors and our man-made structures all over the place. And from the state's perspective, those corals represent far fewer ecological services and functions than corals on natural reefs, so that's what our sources are for these corals. But collecting corals from those environments pose other problems. There's invasive species, there's disease, there's pollutants. So when we bring corals into our nursery, they go immediately into a 1-month quarantine where they're evaluated daily. And in some cases, we even fragment them to much smaller sizes to minimize pollutants and parasites. From that, we then conduct microfragmentation in specialized rooms we have where, I'm giving you a general, we'll target 10 centimeters of source material. We'll break that down into very small 1 centimeter fragments. We get about 140 fragments from 10 centimeters of source material. That is then placed onto, it's cut using a very specialized saw. It's like a tile saw, but it runs salt water through it to minimize stress on the corals. Those are then placed onto prepared modules, which have been soaked for at least 1 month to leach out any chemical issues, and then bleached and cleaned. And then we attach these small fragments using surgical superglue, once again to minimize any tissue interactions, and the surgical superglue will set underwater. Those are then placed in very highly specialized aquariums. These are kind of the equivalent of a 5-star hotel, so they're getting perfect light, very directed light that we control electronically over time. A lot of time and effort spent on water quality, and we also have specialized foods we're giving them. So we're doing everything we can over a short period of time to maximize their growth. And as I said, in the wild, these things are growing 1 to 2 centimeters a year. During this phase in the nursery, we're getting that up to 5 to 6 centimeters a year. So in about 8 months, I can go from having a 10 centimeter source colony to a 42 centimeter module that's ready to put out in the field. And here you see them in our specialized aquarium as they're growing. Here you see one that's almost completely fused over that time period. And that's the key take-home period, take-home message, is that for a place where corals grow so slow, for me to be able to save 20 to 25 years of growth and get all this out in 1 year is a huge gain in ecological services and functions, which we can then use to rationalize the extreme high cost of this program. But there's a problem because I've been raising them in the equivalent of a 5-star hotel. I have to figure out how to get them to be homeless and back out on the street when I put them out on the reef. And so we have a whole acclimation process. It's over a period of 1 month, where basically we have these very large outdoor tanks. We can control sediment in it. We're actually adding sediment in slowly to create turbidity to duplicate the conditions they're going to be outplanted to. We vary the light to recreate the depth they're going to be planted at, and the cloud and light conditions for the reef they're going to be planted at. And we have unidirectional wave makers, so we can create the surge on the reef flat or on the submerged reef that they're going to be exposed to over time. And after a month, we then put them out. And because we're trying to maximize our investment, we're target placing them into, quote, MPAs. These aren't necessarily dedicated MPAs, like a government-run MPA. Some of them are, but there's other types of protected areas. For example, in the US now, if you have an airport near the coastline, there is a security zone that extends out into the water, and those are great places to put corals. The reason you want to put it into a semi-protected area is you're trying to control as many of the human-caused impacts as you can in order to maximize the survival of those organisms against things you can't control. And here's an example of some of our modules that have gone out immediately 1 month after transplantation, and then 9 months later. We're up to 2 years now. That color you see on the right is pretty similar to what we're seeing, very robust, healthy colonies, and especially compared to the same species at the same site that was naturally occurring there. I should mention, we do not introduce new species into new areas in Hawaii, and we're only dealing with native species for what we're doing. That was a native Parietes. This is a native Montipora. I actually have 12 species right now that we're culturing and putting out on the reef with these modules. I have a total of 40 species in the nursery, but most of those are rare, and I'll talk about those in a second. And here's our test spot, and you can see a variety of modules of various ages and various species that have been put out. Right now we're dealing with 42 centimeters, but next year we're moving up to 80 to 1 meter in size modules we're putting out. And the interesting thing about that is it's the same time period. So we'll be producing 82, 84 centimeter to 1 meter wide modules in a single year, from about 20 centimeters to 25 centimeters of donor material. And that represents, in Hawaii, given those growth rates, 150 to 200 years of gain of growth. In addition, as I said, we also at the nursery are maintaining rare species. I have about 28 species that are considered rare or uncommon. Most of them are endemic. Some of them have actually disappeared from their source sites in Hawaii, because a lot of our rare species are in single bays or embayments. 2 of them we've targeted for using this fast growth technique, and we're starting to grow them on small modules. And you can see those small modules there. And after the summer heat maximum, which will be in September this year, we'll be outplanting them back into their original reefs where they disappeared from. This is kind of an overview chart. Kind of gives you a broad view of our program and the benefits and disadvantages of why we're doing certain things the way we are. And then next steps, as I said, are currently, with the size of the nursery I have, it is a land-based nursery. It is very expensive to run. We can produce a maximum of about 80 42 centimeter modules a year. This fall, we're increasing the size of the nursery, and we'll be up to 220 modules a year. Once again, 42 centimeter modules, but we're also creating the ability to go up to 1 meter in that same facility. I'm doubling my staff, so I'm going to be hiring folks within the next month. I'll probably get about 8 people working for me soon. And we're expanding our rare species program. And we also want, I should mention that I just talked about the fast growth, but we have other stuff going on with asexual larvae, sexual larvae, and some other projects. And we are going to be creating a crustose coral and algae project, because we recognize that's critical for our reef survival, in addition to the corals. And finally, just to put this all in perspective, so we're talking here about the effects of climate change and having enough lead time to try to get these programs up in years, perhaps decades, to try to save reefs. All right, this was about a week and a half ago. All right, this is from Fissure 8 on the Big Island. We have an active flow. We had an existing marine protected area at this site that had been there for 20 years. And it was a very, very unique marine protected area because it was a tide pool complex. It was these huge tide pools. Some of them were about 25 feet deep, meters, translated. This is inside that tide pool. So you're looking at corals there that are over 150 years old in a tide pool. And that's why we had made it a marine protected area. This lava flow came through, it wiped out everything, killed everything. We lost an entire MPA in 3 hours because of a lava flow. Never happened before. But you see the circles? 2 years ago, because of the threat of a bleaching event, I had sent one of my technicians and he had collected those corals, fragments of those corals which came into our nursery. So it shows the advantage of doing this for long term, and we may be able to reintroduce that to that island later on. Thank you.